The EpiNext CUT&LUNCH RNA Immunoprecipitation Kit provides a rapid, efficient method for studying in vivo protein-RNA interactions by combining key advantages of traditional RIP and modified cut-and-run, a novel approach to protein-DNA interaction profiling. With this kit, target protein-associated RNA can be enriched within 2 hours. The unique nucleic acid cleavage enzymes ensure low sequence bias, enhancing specificity and minimizing background. The enriched RNA is suitable for both gene-specific and transcriptome-wide analyses. In the assay, cells are permeabilized and treated with a RIP-grade antibody against the protein of interest. A specialized cleavage enzyme removes non-specific RNA-protein complexes, while antibody-bound complexes are retained. The associated RNA fragments are then purified for qRT-PCR or cDNA library construction to analyze protein-RNA interactions. The kit includes all essential components needed to perform the assay. First, prepare the antibody-binding reactions by adding your antibody, the positive control antibody, and the negative control non-immune IgG into separate PCR tubes containing working permeabilization buffer. Then, transfer 20 microliters of your suspended cells into each reaction tube. Transfer another 20 microliters into an empty PCR tube and set it aside. This will serve as your input or no antibody control. Mix and rotate at room temperature for 30 minutes. Briefly centrifuge to pellet the cell debris. Remove the tubes from the centrifuge and transfer the supernatants to fresh tubes. Shake the affinity beads to completely resuspend them, then add the resuspended beads to each tube. Mix and rotate for an additional 20 minutes. Remove the tubes from the rotator, then add RNA cleavage enhancer and cleavage enzyme mix. Mix and rotate for another 5 minutes. Place the tubes on a magnetic stand. Wash each reaction tube 3 times with wash buffer and once with protein digestion buffer. Prepare protein digestion solution by diluting proteinase K with protein digestion buffer. After the final wash, remove the tubes from the magnetic stand and add the prepared protein digestion solution to each reaction tube, including the input control. Mix and incubate at 55 degrees Celsius for 15 minutes in a thermocycler. Place the tubes on the magnetic stand until the solutions are clear. Carefully transfer the solution from each tube into a fresh PCR tube. To each tube, add RNA purification solution, followed by 100% isopropanol. Resuspend the RNA binding beads by vortexing. Add the resuspended beads to each tube and mix thoroughly by pipetting up and down several times. Incubate for 5 minutes at room temperature to allow the RNA to bind to the beads. Then place the tubes on the magnetic stand until the solution is clear. Carefully remove and discard the supernatant. Add freshly prepared 90% ethanol to the tubes, then carefully remove and discard the ethanol. Repeat once for a total of 2 ethanol washes. Resuspend the beads in elution buffer and incubate at room temperature for 5 minutes to release the RNA from the beads. Capture the beads by placing the tubes on the magnetic stand until the solutions are completely clear. Transfer the solution from each tube into a new PCR tube. The enriched RNA can now be stored at -20 degrees Celsius, or directly used to either construct libraries for sequencing or perform a simple qPCR assay. With the EpiNext CUT&LUNCH RNA Immunoprecipitation Kit, you'll achieve fast, sensitive, and specific enrichment of target protein-associated RNA. For more information, visit epigentech.com or contact your local distributor.